So let me recap the 3D comparison. So first of all, the lingo. So from 1D strings to 2D contacts to 3D, three-dimensional view of protein structures. I said 2D contains the entire information in principle except for the mirror image. And the comparison of these 3D structures is to learn about function and to learn about evolution. There are three main databases, one from Alexei Murtsin, SCOP, that shows really how very different structures have similar cores that in fact often confer to similar functions. Janet Thorne, Christina Rengo brought us Cath. Here is a superfamily, so many proteins that have almost the same structure. Here, another example for Cath. And COPS from Manfred Zippel, where we see that we have some sort of similar core here in entities that are very different otherwise. So let's get back to the 1D view of the world, to sequences. And let's first talk about the alignment, the pairwise alignment of protein sequences and how that is done. The typical answer of sequence alignment is how similar are proteins. Why do I want to know? Well, let's begin with the technical examples of how to do it. So you have these two strings, and alignment simply means you define some criterion for what is a good match. Let's just imagine here we defined the identity of letters, and then you shifted it around such that you get for these two strings the optimal match. In this particular case, it's that particular way of aligning them, and this moving around, finding the match, is called the alignment, or aligning them. That's another one. You can get more into it. And now we get into the issue of meaning. So some of you may be familiar with the meaning of this one here. This is father. And now this is something else. Some of you may be familiar with that one. What not all of you may be familiar with is that in fact these three all have the same meaning in three different languages. And it is not related to this one here. Or it is related. It's a grandfather. So it's the father of the father. Clear relation. In terms of meaning then in the alignment, although this all made sense, the sort of meaningful alignment in some sense would only be those three, plus there's some relation here, the father of the father, but this would have been a mistake in the alignment. Okay? Although in principle it was the same way of doing it. And alignments, pairwise alignments, aligning proteins is on the one hand about finding this optimal superposition, on the other hand deciding what is meaningful and what is not. Again, the goal is to align two sequences, and in order to do this we need in fact to do two things, and they're intertwined. The first one is to find the optimal superposition. The second, to in fact define what optimal is. So here I defined as optimal identical letters. So match as many identical letters as you can. That's one particular criterion for optimality. Now, there's a global alignment. In the global alignment, what we want to do is we want to essentially align. Now, this is a real protein sequence here. Protein sequences, by the way, don't have an O. So in a global alignment, we want to, in fact, align all the residues from begin to end. Dots in the alignment essentially mean insertion. So the second sequence here, where that has an LA, the second one has nothing. I'll get back to what that means in a minute. This is opposed to a local alignment. In a local alignment, I don't force the alignment to start at the same position. So I say I could have a mismatch at the beginning here. Okay, the biological meaning of an insertion, in fact, is, let's begin also with the biological meaning of relation of sequences. There's an evolutionary relation. In this evolutionary relation, you may have an additional bump here in that protein. You may have a loop. Let's go to another word, homology. The meaning of the word homology is a very often used word, and homology comes in fact from phylogeny, another one of those words, essentially relatedness. the related origin, and this one here is the only figure in Darwin's 1859 Origin of Species, the tree of life. That's the tree of life that explains how we all are related, according to Darwin in 1859. And the idea of homology, in fact, let's first get into phylogeny, or into different trees. So that's another way of a tree, and you see the three kingdoms here: eukaryotes, archaea, and bacteria. Blue, red, brownish color here. So they're separated. Here is another way of looking at the same tree of life, zoomed into the one that I showed you before. Now the eukaryotes are here. The eukaryotes are here, the prokaryotes are here, the archaea are here, and another zoom in. Between these two different trees, what you see is the way the origin of the parts differs. And this, in fact, has for long been a debate how that is done. Back to the definition, that's the detailed view of that. Back to the definition of homology, or of species. So, speciation event is the point at which you have a common ancestor, and the common ancestor splits into two different species. Okay, that's called speciation. Homology then comes from Greek, homologos, and logos. Homo and logos. Homo is what that says. Logos is what that one says. Originally was introduced by Richard Owen to in fact refer to same organs. So the foot, the flipper in the whale, or something like that. Or the hand. Bless you. In terms of genes, the definition of homology is that we have a gene that originates from a common ancestor before the speciation event. Okay? That's the definition on a genome. So a gene that comes from a common ancestor before speciation. There are two different issues, and I don't really want to get into that. You don't have to learn these names. This is a name that will get back to you, and this is often misused. I'll get back to that in a minute. Orthologs and paralogs. Orthologs, the idea is, so we take this gene, we copy it into a new organism, and in this new organism essentially does the same thing as in the previous common ancestor. So common ancestor has this gene doing function A. The two offsets that are now two different species also do function A with it. In contrast, the paralog is some gene that was copied after speciation. Yeast is a particular example that constantly is an organism that constantly duplicates genes as yeast lives to escape from dangerous conditions, for instance. And in this duplication, they just take a gene, duplicate it, or more than gene, duplicated it, and now you already have the gene that does the function A. With a duplicated version, now you can do something else. And that is one playground for evolution, or for change. And this something else then is called a paralog. So that would be the same gene duplicated, but over time changed. So typically the idea would be that the paralog adopts a new function. Very often, so we have tons of examples that show that. Now, how do you believe species are defined? Sort of, in principle, this picture gives it away.
When they can produce.
Exactly, they can reproduce, they can mate. That's the definition of species, precisely, the biology definition of species. So, you know, the historical example for that story, and I took it here from these wonderful slides. So you have these fruit flies that are happily munching on their favorite food. Then sort of disaster strikes. That is that there is a separation of the two parts of the island through a flood. Now suddenly one species of these fruit flies have to live with a different food because on the other side there are no banana trees by coincidence. So they adopt different strategies and different favors for food and come to the point where in fact these two no longer have the same mating preferences and where then they evolve essentially ultimately into two different species. As biology is always unclear, it is unclear whether these, for instance here, are different species. The similar story is true here. And some of you may know that many of us carry, I guess, I'm not entirely sure. There was one in the room who may not, but that person's not in the room at the moment. So all of us carry Neanderthal sequence in us because, again, the idea is there's Homo sapiens, there's Homo neanderthalis, they are separate species. By that definition they couldn't mate, but obviously biology is not always behaving in the way in which we expect it to behave. So they mated in the past. In fact, most likely mated twice. And there is, so again, the definition is not only mating, the definition is that the offspring has offspring again. So beginning of a line, you correctly defined it, I just simplified it. Again, genes, homology is genes that originate from a common ancestor. Often, this is misused as to say homolog structure. And what it essentially means is similar structure. So proteins that have similar function are often coined homologous, but that is not true. The real definition is through the evolutionary tree. That's the point. The meaning of insertion, as I said, is typically a loop. The evolutionary relation is a common ancestor. Let's get to dynamic programming. Dynamic programming essentially is a brute force. So the way you can align sequences is you can align them visually. Okay? You saw that with these words that I aligned, papa, puppy, and all of that. It takes you no time to do this. For these, you still can do that fairly rapidly. And in fact, there are people out there who can even do this for strings that are hundreds of residues long, and for which you have thousands of examples. There are experts who are really good at visually aligning proteins, and in fact, when we look at the evolution of details and the evolution of viruses, the experts do exactly that. They begin using the type of tools that I will present in this lecture today, but then they do a refinement that is really visually. They print out thousands of sequences, glue them onto boards, and then they rearrange the sequences. That's really how it happens. Needleman-Wunsch is the first way to, in fact, formalize that as into a machine in the 70s, received in 69, sorry. That is the original paper. And the idea is simply you take the first, you want to align the second one into the first, so you slide it along. Right? You try all the possible positions. And to formalize that, simply you write these two sequences out as a matrix. And then you simply put down the numbers. So our criterion for optimality here again is matching letters, identical letters. So then we fill out the entire matrix. So if I want to align the first of the second sequence here, the E, against the first one, the first time I find a hit is here. The other hit I could find is here. Now once I find a hit, aligning means that the next step here would be to take the next sequence, right? Or the next residue. So that would be E on 2 against E on 1, G on 2 against E of 2. The score still is 1. So you see that I had a hit on the first, but I never find another hit. An identical letter. We could start with a G here. Again, I still get a score of 1. We could start with a G here. Now, in fact, for the first time, in fact, I get two hits. And that essentially is the best you can do if you don't allow anything else. And that something else that you can allow are gaps. Gaps essentially allow you to jump in that direction. So not move on the diagonal, but just to insert a point. I showed you that on the slides before. Inserting a gap here would, for instance, imply you start with a G here, go forward, and then insert a gap. So far the score is 2, and by inserting a gap, you in fact increase that sum to 3. Similar one here, another gap, and some of my smileys are gone. So the optimal score here then would be 4, and then you backtrace. And you have the optimal alignment, backtrace. It's not that simple. I sort of omitted a couple of steps, but that's the principal idea. The principal idea is you have a formal way to try all possible combinations. As long as you align the sequences in a global way means from end to end, This is mathematically proven to give you the optimal alignment. Again, it depends on your criterion for optimal. Here, my criterion for optimal is identical letters. Whatever your criterion for optimal is, this is guaranteed to give you the optimal solution. Okay? Wait, is a very, very important assumption that I have been dropping here. As long as your criterion for optimality does not require a dependence, the alignment for this A is not allowed to be dependent on the alignment for the Q. Put differently, I assume that what I do at the Q is completely independent of what I do elsewhere. Okay? Which in reality is not true, but which in fact is being used today for essentially all majorly used alignment methods. Okay, so again, as long as I define any criterion for which the optimality is defined only for a single letter, any definition of optimality will be optimized by this global Needleman-Wunsch. Overall, this is a larger protein, and you can plot these dot plots where you see sort of some density, and when you see this density, somewhere you see that there are regions where you have more density, so where you have other alignments that could be the case, could be good. Now we get into the next issue. What is best? So assume now we allowed some local alignment. The question would be, is it better to have 2 residues aligned with a score of 2, so 100% sequence identity, 2 and 2, or is it better to have, in this particular example, 5 residues aligned? No, is 2 plus 5 7 residues aligned with a score of 4. So we no longer have 100%, we have something in the order of 50%, but we align many more residues. What is better? Here's another issue that we have to handle and that I oppressed so far, that issue of the gaps. So as long as I have a linear gap penalty, so gap penalty essentially means how I penalize the fact that I don't align a letter here, these 2 alignments are completely the same cost or the same value, right? Letter, gap, letter, gap, letter, gap. I said that the gap, the idea of a gap is you have a little bit of a bump. So this would correspond to something bump, something bump, something bump. And that is, as already hear me talking, unlikely. This is the much more likely event. And that brings us to the idea of blocks. That idea comes from Stephen Henikoff originally. So when we look at the alignments of many, many, many proteins, typically we see something like this. You may remember my talk about domains. I showed things like that. This is one protein, another protein, 3, 4, 5, 6, 7, 8, 9, 10 or something like that. And this looks, so you have a region here that is aligned, a region here that is aligned. In between you have a gap, and this in fact looks like you have 2 domains. Domain 1, domain 2. That's in my 3rd lecture, I believe I sort of used that in fact to digest domains or dissect domains. The idea of the blocks is simply to look at alignments and to find these blocks and to learn from them. Now, how could we enforce blocks into the alignment methods? The idea is we in fact treat differently whether we open a gap, so the first one here gets a different penalty than the extension of a gap, the second one. And typically the extension of a gap is 10 times lower penalty. So this way you guarantee that you can in fact not easily open a gap, but once you have it open, you sort of can relatively easily continue keeping it. This simple function here gets you to something like that. Now again, the optimal alignment from Needleman-Wunsch needs to be global. I've put that under the carpet. I showed you a couple of local alignments already. The local version of the same dynamic programming comes from Temple Smith and Michael Waterman. And again, so these are local alignments that are allowed for bless you, for this. So this is, for instance, an example of a local alignment. And this, in fact, would be the gap elongation, the gap open, penalty, and the entire alignment score. You simply sum over all the hits. That's as simple as it is. Other than that, you do the same algorithm that I showed you before with the same omission of the details. Yes?
Yeah. So the question, because in all of this way to compare sequences, we assume that there is a linear order, but what happens if we have one group of letters that have a periodic repetition?
So the question is, we have in all of what I presented to you so far, we assume that we have a linear order. What if this assumption is incorrect? And let me just paraphrase your question and answer another question first, which is, what if I inverted the sequence? I would not find that. So if there would be a similarity of the protein in that direction, I could not find that. That's the first thing I wouldn't find. Since we cannot find it, we say we haven't seen it very often. Okay? Which, as you see, is sort of a circle. We assume it doesn't exist often, but, you know, could be. The question that you asked, what about local motifs or local repeats? In fact, that was exactly the question. So, local repeats, what I now show here is the local alignment. The local alignment then should sort of hit its maximum when it gets to the local repeated region. So assume you have repeats of 5. 5, 5, 5, 5. Then actually aligning those 5 should give you this top score. Bless you. And what's happening today? It's infective somehow. But in principle, that is the answer. That is the solution is built into the system, and it's all easy. But the first thing I already asked before, what is better 2 in 2 or 4 in 7? And so it's not that clear how to get there, and that leads us over to, in fact, finding repeats is typically done by different types of methods. Because these methods, in principle, they could do it, and for long repeats, they, in fact, manage to do this, where long is defined as 50 residues. But for shorter repeats, they fail. And that's a major issue, and there's a whole generation of other methods out there that focus on motifs. It's highly non-trivial, and we get to the reason why that is the case. The first question that I want to ask, is there any other question, by the way? There was some discussion here in the front. No. So the first question I want to ask, is identity in fact the best way to define optimal alignments? And that brings us to the substitution matrix, and that brings us to, obviously the answer is no. There are 2 different potassium channels here from 2 different organisms. When we align them in the right way, we see in detail, if I gave you the time to read this in more detail, you would see that there is a lot of difference here. In fact, this row here shows the identical residues, and you see there's a lot of white, a lot of difference. The functionally important region here, the sensor of the potassium channel, are these 3 residues. And you may see that in these 3 residues, only one, in fact, is identical. So we have 2 proteins that essentially have a similar structure, that essentially have a similar function, and one very, very important aspect of that function of this potassium channel is this highlighted in yellow region, where you have only 1 and 3 identical. So immediately that tells you that letter identity is not necessarily the best way. What else could we do, and why could that be the case? Let's begin with this, with Margaret Dayhoff. Margaret Dayhoff was one of the first people who had the idea of really understanding how evolution works by comparing sequences visually. She compared thousands, she compiled a compendium published in the first introduced in 78, but there is a publication in the 70s that I'm missing out here. 1572 mutations in 71 families. So she simply visually aligned all these proteins and tried to read off the evolutionary event. So simply, what can we change in evolution? Which residues can change? Where? How? And she defined point-accepted mutations as a substitution matrix that simply gives the probability of substituting a particular letter at a particular position. Where PAM1 is you have 1 point mutation in 100 comparisons, and PAM2 is by the power of 2. Okay? PEM 20 would be by the power of the PEM 1 by the power of 20. So it's not 20 substitutions. Here is a sort of next generation way of doing this, done by Stephen Henikoff and Jorge Henikoff, his wife. It's the blossom matrix. The blossom matrix, again, Stephen is also brought the idea of the blocks. The idea is find these blocks, and then in those blocks simply compile log odds. So probability of change with respect to the background probability. You know the frequency of your letters in the alphabet. That gives you a simple background probability. And then you see how surprising is that substitution at that particular position with respect to the background. So again, this starts with blocks. Blocks are defined as things that have no insertion. Okay? We have regions where we not have all the same residues, but we essentially don't have insertions. These blocks can be 10 residues long, they can be 20 residues long. That, I believe they scale between, the average is in the ballpark between 10 and 20. They can be between 5 and 40 residues long. Again, no insertion in this, many proteins aligned, and then we simply compile the log odds. That gives me a matrix where I have the 20 amino acids here and here, and then I look up what's the score of E on E, that's 4. What's the score of A on I is -1, so that is unfavorable. You don't want to do the alignment. Let me get back to that. I thought I had it. So here's another point of that. The diagonal is always scored to itself. It's the match of the letter. And you may see, I'm not sure, I'm sorry for the quality, I copied that out of the paper, which was on the first slide, but now I dropped it. I'm sorry for that. So you see that the diagonal is not always the same. So here the diagonal is 17, while here the diagonal is 2. So matching two A's, alanines, is not as important as matching two W's, tryptophans. Because tryptophan is very important for function. That's why this is a much more surprising, much more evident of a biological event underneath. And that's why the diagonal here is not identical. And that immediately gives away how important it is to have something other than sequence identity for the alignment. In fact, you see also that matching an L on an F gives the same score as alanine on alanine. And that is not an identity. Another thing that you may see is the I on the L, for instance, is in a good score. The I on the V, isoleucine and valine, those are both hydrophobic residues. So essentially what you match is the biochemical feature. And there you have immediately the explanation. The reason why these matrices are important, why Blossom is so important, is because it sort of tells you something about the biochemistry of the process. Sometimes it's not important that you have an isoleucine there or an L for that matter, but it's important that you have something that's hydrophobic. Everything else is unimportant. And yes?
How exactly did they come up with these numbers?
So the way they come up with the numbers, Stephen and Jorge, again, they compile a set of alignments, of blocks. So they take a protein, align another protein into it, align another protein into it, align another protein into it, find all the blocks defined as no insertion, take this block, and for every column in this block, at position 2, so the second residue, what do I see? Do I see only alanines? And then I, in fact, in this way, compile the log odds for the observation of alanine in that position. And you do that over all the blocks in a set of 100 proteins. Initially, I believe they started with something that may have been in the ballpark of 100. Is that clear? There are many, many more matrices. The 80s and the 90s were the 2 decades of defining matrices. McLaughlin, Claverie, Gonnet are other. So the one I talked about is Blossom 62. 62, there are other blossoms that are derived in the same way. The number here simply differentiates how you select the first set of sequences for which you define the blocks. 62 means that everything in there is 62% sequence identical. And here we are another circle. So you understand that percentage sequence identity matching the letter is not the best thing, but you choose exactly that criterion before you define a substitution matrix. You don't have anything better. So you begin with percentage sequence identity here. That's the 60, where the 62 comes from. And then there's a 30, blossom 35 for 35% and so forth. Then there are structure-based matrices, fat and slim, as you may suspect, sort of are competitors to each other, and they both have to do with membrane. They try to see what you can change when you have protein inserted in the membrane, and there are other structure-related functions, exchange matrices here. Again, for all of these, the way they are compiled is different. Typically, they all compile log odds. What they differ in, mostly, there are other differences, but mostly what they differ in is how they collect the first set of sequences for which they compile the log odds. Okay? Gonnet does the log odds also differently, McLaughlin as well. But mostly, the most important difference really is in the initial choice of the data set. And sometimes this initial set of data is chosen by function, meaning instead, I said here, you find every protein that is sort of has a region in the membrane. That is the criterion for selection. Here, you find everything that has a particular function, or the like. And there are many, many, many others. By the way, there's a nice sort of interactive tool from Francisco Melo, interactive tool to play around with dynamic programming if you want to. Here is the link. A big problem in dynamic programming is the time that is used, so it's slow, and it's unclear how to choose the parameters. So I said there's a gap opening, there's a gap elongation, there's a particular matrix, those are free parameters. How do you choose them? And there is no optimal choice for any of those. So that implies again that yes, there's a default with which the programs come, but typically these programs are written by those like you who have a background in computer science or a background in math or physics and do not know at the beginning of their career very much about biology, and they write a program that simply finds an optimal solution. But they are meant for people who know much more about the biology and they know how to set these parameters. That is one background idea in the alignment field. Now, here's another idea to speed it up. So speed is an important issue, and I should have brought you a different slide here. In 2009, let's just put these numbers down. I'm not sure that everybody can see it, but I put them down, I believe, more to write something. So in 2009, when I came to Munich, we had about 6 million sequences in UniProt. Now in 2014, as I said, in 04, that is 09 here, we have 55 million. So over 55 months, this is an increase of more than twofold per every 2 years, okay? That's the number of sequences. But for the alignments, what you in fact have to do is a pairwise comparison. Meaning you have to do the 36 here and the 3,000 something here. 3,025 million. That's a sort of much, much bigger increase. And in fact, not only is that a much bigger increase, it's a much bigger increase than in terms of CPU. So what we had to do when we came to Munich and what we have to do today, we could more easily do it then than we can do it today, although the hardware and everything else is much cheaper and much better today. But it's not good enough because the biodata grows further. And again, the problem really is that everything starts with sequence comparisons, and sequence comparisons essentially square the problem of the fact that the biodata is growing so quickly. So we need to speed up. And all of this problem is after the speed up, but still, I'm using only this example to show how important the speed up is. Speed up to dynamic programming, fast and dirty. And one way to do it fast and dirty is by hashing. You simply look at a word, you begin in the first step, you match words. Very, very easy to find strings that are identical. So again, in some sense, you could imagine this to be a short repeat. Now, assume you take these three words. You have a word size of 3, and you simply find everything in this pair of proteins that has identical 3 letters, or words of 3 letters. Okay? Now what next? So in this particular example, I would give you that. Okay, that's already the answer. Sorry, I saw my slide incorrectly. So in the next step, you try to extend this first match in either of those 2 directions. How could you do that? How could you try to extend? Alignment. So by the dynamic programming that I did before. That is a proposal on the table, and it's a fair proposal. That's all you know so far. So, but I'm talking about a method that is supposed to be much faster. Now the idea is, okay, my word matching in the beginning was very fast, but now I extend and I use the same dynamic programming, then I'm very slow again, right? Wouldn't that be a contradiction? The answer is it's not a contradiction, but tell me why. In fact, that is exactly the way it's done today. Initially it was not done that way, but today is the way it's done. Why can that be done? So why is dynamic programming possible? The fact is that dynamic programming the speed is in the order of the length square. Okay? And that is in fact exactly what helps us here. By breaking the problem down into small bits, instead of aligning from here to here and have that, I cannot remember, maybe 80 residues. So in fact of squaring 80 here, I square 10. And 80 square and 10 square are very, very different numbers. That's why you can get away with doing the the dynamic programming for filling in the regions in between. But still, this may lead to a situation in which in between you do not know how to proceed. You sort of are left hanging, meaning you don't know whether there really is a very, very long insertion in between or not. There are 2 essential tools that do something like that. The first one historically was FastA, proposed by Bill Pearson. And the second one is BLAST, Basic Local Alignment Search, initially by David Lippman, NCBI director, also Stephen Altschul, Webb Miller, Eugene Myers, grep. Some of you may sort of know Unix or Linux grep, that is also from Eugene Myers. Tom Madden and Alejandro Schäfers. Again, this is 5 years before that. This one here, they differ in fact in the statistics. I'll get back to that in a minute. The major problem here, and that is exactly the same problem that you also have for finding repeats, the major problem is to look at things that are statistically important. So the shorter the word size gets, or the shorter in fact your alignment gets, the more likely this hit could have come from by random. We have 55 million sequences. If you just randomly created 55 million sequences, I'm not entirely sure about my statement now, but most likely you would have any imaginable 3 letter size in there. Yes, clearly, it's 20 by 20 by 20. Probability is very, very high that 55 million is much larger than 8,000. So the point is that in just a random subset or in a randomly created universe would match 3 letters is so high that in fact a match of 3 is not enough. It's not statistically significantly enough. And that is the problem the shorter you get. And repeats, when they are short, that's exactly the type of problem that they have to fight against. Now, again, the idea of statistically significant is you look at some background that you could take by randomly shuffling sequences. You know the overall amino acid composition in a database of 55 million, and you randomly shuffle. Or you could take it by simply taking the database and going through it, and you look at the score of the things that you actually find. And I should rename this here into HIT. And the statistical significance, in fact, is a function of two points here. It has to do with how wide this curve is, or 3 points, where its average sits, and how far this differs from that curve. That defines statistical significance. And again, the big difference here between fast A and BLAST initially was that fast A really did essential permutations. This is much better statistics. BLAST compiled an overall propensity for the overall composition of amino acids. This is way faster. This was better. Meanwhile, since BLAST has improved, now is faster and better. Any questions so far? That would, for me, end the idea of pairwise sequence alignments. What I want to tell you or make clear here, I summarized essentially 40 years of research, and that's one of the most active fields of research in computational biology, comparing pairs of sequences in just a few slides. But the main ideas really, I'm going to give you more ideas that have to do with more than 2 sequences later. But there are any tool that you could imagine, machine learning, statistics, has been applied to align sequences. I just showed you the 2 main ideas that in fact are most powerful in the sense they are mostly used in the field. Dynamic programming and hashing. And for hashing, BLAST is the tool of the day. Now, yeah?
Would we first run BLAST to find potential matches and then would run dynamic programming to make the solution better? Or?
Yes, so the question is, I essentially said, implicitly, I said that dynamic programming is better, BLAST is faster, so your colleague asked, why not first run BLAST, and then after you find a set of hits, so we have 55 million, you run a fast BLAST through the database, maybe you assume that in your family there are 10 members. And what you do in this fast BLAST, you find 1,000. And then you do only 1 against 1,000 instead of 1 against 55 million by dynamic programming. That's precisely what people have been doing. Great idea. It works exactly like that. Today, and I'll get back to this issue later in the talk today or next week. There are, in fact, other ways of approaching this. I'm completely confused. Oh. Okay. How accurate are pairwise alignments? Let's begin with a few statements. A similar sequence implies a similar function. Is that true? I'm not seeing a lot of. Who in the room feels this is not true? It's 1, yes, 3, 2, 3. Yes, it's true, sometimes it's not true. But mostly, in fact, it's true. Mostly, this is, in fact, exactly the principle of evolution. Homology I defined is you have an ancestral sequence that does the same in the different organism. Sometimes you have slight changes, but you still do the same thing. But there are many examples where it's not true. True. Similar sequence, sorry for the typo here, similar sequence implies a similar structure. Is anybody against that or believes that is wrong? So no hand anymore, one dares. Thank you. So this, in fact, so there were 3 here, 1 here, and that is also true. So there are examples where, in fact, similar sequence does not imply similar structure, but this is a stronger statement than that. More often, similar sequence implies similar structure than it implies similarity in function. So the group has, as a group, chosen the right thing. Let's just look at the problem in terms of space. So assume this is the space of all the sequences, and this were the space of all the structures. Again, the same sequence essentially does the same structure. Different sequences, in fact, can come close in structure, can have similar structures. Slightly different sequences mostly have similar structures. Most similar sequences will not have any structure. Most similar sequences, at least today, have not been observed. Most possible sequences, sorry. So what I mean here by these dots is not similarity in terms of between mouse and human. What I mean here is I just randomly create sequences. And then most values in between, most changes have not been observed. So for what I talked about, there is a zone in which the alignment is relatively straightforward. And typically people refer to that as a safe zone. Sometimes you can sort of map it in terms of pairwise sequence identity. I said one of the Blossom 62 is sort of 62%. And since they started with Blossom 62, that sort of gives you an idea that they believe that 62 is a fairly safe point to in fact align sequences. It's relatively easy to do this there for pairs. It's a little bit tougher to argue for that, but there is some, wherever this zone ends here, there's some safe zone for the alignment. And then there's a twilight zone, and that term was coined by Russell Doolittle. So a zone at which you sort of lose the signal. Interestingly enough, where that comes, there are two ways in which you sort of could find out where that comes. One is you could just align things for which you know some similarity. For instance, you could take all the things that have similar structure and see what the sequence similarity for those is. Okay? And then that would sort of tell you where you lose the signal. Another way in which you can do that is you just randomly create letters and compile the probability that if sequences were random words that had nothing to do with evolution, you still could define the point at which you would sort of lose the signal. And they are very, very similar. The points where they meet, this depends a little bit on the detailed assumptions, but actually why we go from safe to twilight has to do with biophysics, biochemistry, has also to do with simple sequence features. Not protein sequence, just sequence as sequence, the comparison of sequences. And then there is a midnight zone. So the idea of a midnight zone is that now, so here you somehow still can see that the two things are related. It's not very clear to see that. In the midnight zone you cannot see that at all. So there, the match, similar structure for instance, looks exactly the same in terms of sequence comparison to the non-match, not similar structures. And I'll get back to that issue later. To illustrate, and this is not true, I'll show you the true data in just a moment, but I want to illustrate the concept of the twilight zone here. What I plot is the accuracy of findings. So accuracy findings could be, I want to match proteins that have similar structures, and I say I make a mistake if the two proteins that I match do not have the same 3D structure. So at some level of sequence identity, I said 40% is safe zone here, I don't make a mistake. So that definition of safe zone is 100% right. And then sort of I drop, okay? Now, the problem is that at some point here where I drop in my accuracy, I also lose things that I wanted to find. So I have a payoff. And that brings us back to the question, how can we in fact assess alignment methods? So I told you that Blossom was developed by simply running the same, using a simple dynamic programming to run at 62% to get a substitution matrix that is allowing you to do better alignments. Okay, that's somehow a circle. Now, is there a way to assess how good alignment methods work without going into a circle like that? Does anybody have an idea? And in fact, the way has to do with something that I've already introduced in one of my previous lectures. Any idea? So let's just play, try to play something. Let's call it an experiment. You form groups, somehow local. I mean, I immediately see this is not possible for everybody, but many of you can sort of form local groups for 1 or 2 minutes and discuss and come up with a proposal. I give you 2 minutes, and you all discuss with your neighbors. Okay. Anybody has any suggestion how we could do this? Any group? Yeah?
Maybe I'm not 100% sure if I understood the problem correctly, but we have the sequence, we know the function. That's what we know. And then we match the second sequence. Then we propose the same function, and then we check if it's the same function. And we do this multiple times, and then check for the accuracy.
So, but how do I know the real answer?
I don't know how to check. I mean,
so we have a solution here. Let's say that it's not completely complete. So the solution says, I take a protein, I find a protein that I match it to. Once I've matched these two, I ask, do they have the same function, yes or no? And I do that many, many times. That means for many, many proteins. The part that is not quite complete is that he says, I don't know how to match the function. We could have an experiment. So we could look up all the cases for which you have a similar function. Any other idea?
Can we ask a human expert if they have the same function?
So the question is, could we ask a human expert whether they have the same function? It's a related question. Typically this human expert would be somebody who has looked up PubMed or experimental information for known function. So in some sense, a human expert in most cases would be somebody who sort of uses databases today. For these cases at least, depends on how many you want. For one family, we could, there may be human experts who are better experts on that family than the databases. If you wanted to have 1,000 different families, most likely human experts are not the right solution for that in most cases. There was a point over there.
Mathematically or theoretically, we can calculate the highest attainable, highest accuracy. Let's say, and then we calculate all the other accuracies and then check the standard deviation from the highest and assume that.
But how would you get the highest possible? Did everybody understand what he said? So I believe that nobody's further away from him than I. So if the acoustic of the room is not unidirectional, then so how do you get the highest possible? I would argue the highest possible accuracy is 100%. Now what do you do next?
Highest possible accuracy theoretically in this particular, from all the 55 million.
But how do you get it?
That BLAST or whatever that is.
No, but how does that give you an accuracy? So BLAST gives you a similarity in sequence.
Yes.
It does not say whether BLAST is right. What we try to achieve, let me repeat, the task is to see how good our alignment method is. So we have an alignment method, we create results, and we want to find a way to verify that what I found is right. So here are two approaches that say use similarity in function. Let's assume that this is what you mean. So optimally, no, it's not what you mean. Then tell me what you mean.
The solutions that are being put by the other students, they take, for example, like accuracy as a local variable. They take what accuracy is available and they judge in terms of that. So what I'm proposing is taking it into a global perspective. But to have the global perspective, you need to have a theoretical higher accuracy.
Okay, let me turn that around. There's a much larger audience here. I'm one, you are many. Does anybody understand what he says?
I think I understand, but it's the same.
No, that cannot be true. So he says he understands, but he says it's the same. And since you explicitly said it's not the same, that cannot be the...
I mean, he proposed that matching two sequences, I already know the accuracy for some cases. And then I calculate from that. And he's saying that after somehow we calculate the accuracy based on some rules that we know already. For the
So he understood you. But you know where I lose you is he also says somehow. That's the part that I'm asking the question for.
When he says somehow, that's what I mean by theoretical high accuracy.
What is theoretically higher accuracy?
That is a variable that's sort of an unknown variable in this perspective.
That is tried, but I ask you how to make it known.
That is, I don't know.
Oh, but that was the question. Okay, now let's repeat. That is the question. So the question is really, how can we check out what is the accuracy that we achieve for every given pair? Again, we have solution on function, yes?
We can try more than one times. Like you can.
That's what they did. So she's, yeah? You have something else?
No, I haven't quite understand that.
Oh, you haven't understood the problem?
I understood.
Yeah. So she said, just do it more than once. So look at similarity in function, but that again is clearly, yes. So one pair is not enough. You do an alignment of a pair of proteins, you look up their similarity in function, and you do that many, many, many times for many, many, many different families, for many, many different proteins, and that gives you accuracy.
I would like to extend this comment that, like, the other guy proposed that if you have been able to assess the function of one protein, then you will compare it with the other protein. So that's how you will repeat the same function with other proteins. You will get a higher accuracy kind of data.
No, again, so we do a pair, we assess the function, we do another pair. I never said they have the same function. In fact, he said, could we ask an expert? We can only ask an expert as long as it's the same function. So by, in some sense, answering that question, my point was, no, we're going to use many, 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 many functions.
Yeah,
Okay?
I would like to extend this comment that what if you create a hypothesis to create some kind of variable or the figure, and then you compare with those figures with the other protein sequences. So that's how you will get to the...
I'm still unclear how you... So I'm not entirely clear where you're going. So the two of you assume that what you were thinking about is sort of a random hit. So I could compare, so I could align my real sequences and could compare to random. In some sense, that's something that we had for the repetition example. And that would be one way of defining, let's call it a theoretical accuracy, because I would compare it to something that I know is not reality. And that, in some sense, you could call theoretical. I would follow you on that. And that, in fact, but this, ultimately, if you think through, is statistical significance. This is exactly what we have been using in fast A and blast. So that's already out of the door. We need it. You're totally right. That is what we need. But now the question is, having used that tool, how good are we? How good does it work? This is science. We can actually measure. And one way would be function. Anybody else has another way? Guys, this whole lecture is sort of, on the title at least, about protein structures. And the comparison of protein function I have not presented to you yet. What I have presented to you is the comparison of structures. So we could simply take a sequence, a bunch of proteins from the PDB, from the set of proteins for which we know the 3D structures, align them in sequence, and align them in 3D structure. Those are the two methods I presented to you so far. This morning I had a recap that you can compare 3D structures and you can identify similarities in 3D. Today we talked about similarities in 1D. Let's assume this is the truth. We call that the truth. And we don't actually, again, we could use experts, we could use SCOP, we could use any of these databases. Through the 3D structural alignment. There is an additional minor complication here. So there are cases where I really know it's a 3D structure is similar. There are cases here where I really know they are different. In between there is some sort of gray zone. All those pairs I can throw away, right? I have enough pairs. So I'm only going to look at these, the right ones, and these, the wrong ones. And now I can assess. And I can do an all against all. It's not that simple, and we will get back to that in more detail later in the lecture, is an essential and important step. And that is about sequence-unique subsets. Part of that has to do with the way biology evolves. I said that a protein structure, to do experimentally a protein structure, costs about $1 million. Now, once you have one, to do another one for a very, very similar protein is cheaper. Okay? But still, so somebody does a structure for the potassium channel, gets a Nobel Prize for that. So the incentive that now you do another one, very, very similar one in mouse, or no, that one was bacterial, in another bacterium, is very high. It will not give you a Nobel Prize, it will not give you a science publication, but if you achieved it, it will still give you a very, very high publication, because it continues to be an important structure. Very hot one. So that is one reason why, in fact, today's database is highly biased by things that are very similar. You may remember in the last lecture I briefly had the slide up from Manfred Zippel's group in which they asked how much novelty is there every week. And there was very little novelty in some sort of 3D structure. One reason is that we have so many structures already known. And the ones that we don't know are very, very tough to do. Another reason is it's much simpler to repeat something that is somehow similar to what somebody else does. Okay? So this is why we really have to find a sequence-unique subset. If we took the whole thing, we would sort of use a bias that has to do with, let's call it, social events. Not with science. Not with a distribution that I have about proteins in our cell or wherever. We take this unique subset and align everyone in here against these. Okay? And then again, I measure. And that brings us to a curve. I should have had the faces here. That was initially published by Chrysander and Reinhard Schneider. What I show here is number of residues aligned, percentage sequence identity from 20 to 100. Essentially, you see they saw this, postulated this curve here, and the curve is such that the pairs that have similar structure are above there. Or put it differently, above you do not find pairs of proteins that do not have a similar structure. The curve was refined a little bit later on because it turned out when there was more data that it had to be slightly refined, but in principle it's sort of the same idea. The level of sequence identity that you need to align longer is much lower than what you need to have shorter, and there's a point here for 11 residues. You have 11 identical residues. That doesn't tell you anything about structure, because you see them in very different, in an 11-mer, an octamer, you see not 11. An 11-mer you see in many different structural environments. Okay, so here the raw data. That's the gray, the curve that I showed you. Blue, pairs of proteins that have similar structure. Red, proteins that do not have similar structure. So you see that what I showed you, what I said before, is not quite true. You have some red above. And importantly, there's some blue below. But clearly, this line somehow separates out blue and red. What you don't see on this slide, really, I have to, before I go on, make a point. Under here, there's a big issue, there's a big unknown here that has to do with the way this is done. What you don't see is the density, but I'll get back to that in a minute. Let's get to this example that I showed before. This alignment here, oops, I took the animation out. Okay. Oh, sorry for that. The first way of correctly aligning these two was in the blue region here. The second way of incorrectly aligning them is in this region here. That's the major point. But now let's get back to the question about, in fact, the numbers, how the density of the points that you couldn't see is really. What I'm also going to show you now is curves that show you results as a distance from this curve. So 0 distance, minus 5, plus 5, and so forth. Okay? Now, that's the percentage sequence identity that somehow relates to that for long alignments. That's the actual distance, that is the measure. That's the insertion or the intrusion into the twilight zone. So you go from relatively few false positives to relatively many. In fact, upon intruding into the twilight zone here, so let's begin with the first point. There are some red points. So this is the number at the curve. You have some mistakes. Below the curve or above the curve, you have also mistakes, but relatively few. Getting to this curve here, it goes up by more than an order of magnitude, the number of mistakes. Now, I'm sorry. I'm sorry. I made a mistake. Blue is not the mistakes. Blue is the ones that you want to find. Blue are the ones that have similar structures. Okay? The mistakes are in red. I'm totally off. Below you don't have essentially mistakes. The mistakes go... The important point, I'm sorry, I gobbled this one up here, totally. The important message here, mistakes in red, logarithmic scale, correct pairs of proteins that have similar structure that you want to find in blue. Both go up in the intrusion in the twilight zone, which is sort of defined by this 0 line here, above the twilight zone, below the twilight zone. Both go up, but the wrong ones go up by 5 orders of magnitude. The true ones go up by 1 order of magnitude. That is defining the twilight zone. That's defining why this is so important to intrude, because this goes up by an order of magnitude. And why it's so difficult, because this goes up by 5 orders of magnitude. That's the challenge of alignments. Okay? Of pairwise alignments. Now, if you only used percentage sequence identity, and many people in the world still do that, then you would start out with 10% and would fall below. So that really is a bad idea to do. Now, overall, in terms of the accuracy versus coverage, accuracy is how many of the proteins that you find are correct. Coverage, how many of the ones that you want to find you do find. Ideally, you want to be somewhere here. And pairwise alignment methods are here. So again, the dumbest thing to do is to use sequence identity only with Smith-Waterman or whatever you do, and you're down here. The cleverest thing is to use the raw alignment score or this HSSP curve that I showed you before, and that's sort of the best thing that you can get. But it's way different from this. So as long as the pairs are very similar, we can do it. As soon as they're no longer this similar, for pairwise alignments, we have a serious problem. That's essentially the message here. Now, for pairwise alignments, we have a problem, I said. What else could we do? What is the next step up if I'm not looking at pairwise? Well, let's begin with a sort of hack, pairwise hack, the triangular equality. Let's assume that a circle defines everything I can reach in a pairwise by some level of acceptable mistake. Say I want to find everything that has at least 80% accuracy according to the curves that I showed you, can look up where I have to stop my alignment. I start with a protein in the center that essentially defines a circle around that center of what I could find safe in this 80% accuracy zone. I can do the same for protein B and C. By my definition of this circle, the 80%, A and B and C cannot be related to each other. Okay? Now let's assume we had a protein here that is in between. So it's in the safe zone of A, it's in the safe zone of B. How could I use that? What would that imply?
Getting the sequences. Getting the sequences from the one that's in middle, the X. So getting that's for both of them, for protein.
Yeah, exactly.
The sequences.
So again, let me just repeat. Protein A finds a related sequence X in the safe zone, so X has the same structure as I, and B finds also X. So B is the same as X. What does it mean? X and A and B are similar. Yes, exactly. And when we do that, here, that is way below the twilight zone. Remember, that's the twilight zone at 0. The blue line, doing this in the right way, you remain at 100%. You do not always find these cases. In fact, you don't find them very often. But when you find them, this is the right way to do. Now, the next step up from here is the comparison of multiple alignments. So we have 12 minutes left, and I could, I guess, run through the remaining 12 slides and introduce the idea of multiple alignment, or I could open the floor for questions. Which should I do?
About the graphics before this slide. We have one horizontal and one vertical numbers.
So I believe the question here is very, I'm beginning to the first part of the question, what does the axis mean?
Yeah, the axis, yeah, exactly.
So, and for that I got a little bit fast because I made a mistake, and that made me speed up. And that's not good. So, where is the idea of the distance introduced? Here. So again, this is the number of residues aligned, percentage sequence identity, that's the curve, the H-val. So now I'm going to define distance from this curve, where +5, +10, blah, 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 blah. If you put yourself on a particular length, you sort of see that at 250 roundabout, this curve sort of tethers out, is saturated, right? That means you could look at 250 and translate this to corresponding pairwise sequence identity. Okay? The translation here is obviously true only for a particular point, while the distance itself is true for all the points. And that is the two axes that I use here. That's the real one, and that's just for people in a translation. That's not the real one. That's the way the data is collected. This is, in this particular case, number of pairs. In the final slide for which you asked the question of the intermediate sequences here, the axis simply is cumulative true pairs. True pairs, again, meaning that every pair of proteins has a similar structure. A pair of protein found by that method. And again, the method uses simply, once I find this pair in between, the question is at which level is that to A and at which level is that to B. Now I merge the families, and am I right or wrong in doing that? And that's the blue curve. So essentially I'm always right in doing that, even at very, very low levels of sequence identity. And let's just repeat this point here, the 0 point, where did I have the HSP curve? There is a cumulative accuracy here. The 0 at the 0 point it's already much lower, and at the 10 point you see that the overall accuracy is dropping to below 20%, and that is where this curve remained. Throughout this curve it remained at 100. That shows you how much better it really is, but it is not applicable to many cases. Any other question? And I believe with that question you sort of almost nailed the answer to my question. So we need another question. We have 9 minutes, which is no longer enough for the next 12 slides. I could do it. I could still do it. Oh, there is another question. Okay. Multiple alignments. I'm still open for question. The first way of doing multiple alignments is simply to use the same old dynamic programming, but now add a new dimension. So do what we did here my plot is a hack, okay? So this would have to be 3D. Essentially, do a 3D of a dynamic programming. There is a recent publication here, relatively recent for this idea. The idea is much older than that. Again, dynamic programming comes from the 70s, so in that sense this is a recent publication here that establishes this of 3, for 3 dimensions, to be an NP-complete problem. Don't have to tell you, this means stay away from it. There is a sort of a claim that there is a computer hack solution for dimensions up to 6. And this, for a short protein, takes 60 terabytes. I just heard that in a seminar. Do you know what the quote was? No. I have to look up the quote and didn't have the time. So for one set of proteins, so for 6 proteins of 300 residues, this would need a computer with 60 terabyte of main memory. And would take a long, long time. So depending on where you're from or how you look at the world, you may call that solution found or undoable. But that brings us to the couple of hacks. The first hack is pairwise, but you somehow look only at the space that is intersecting, that is close, in close proximity. And that comes from Lippmann originally. The second hack here is mapped to a tree, and that comes from Russell Doolittle. And the idea is the following. So you do a set of 4 sequences here, pairwise alignment all against all the pairs. Take the matrix, and in this matrix you write down all the pair relations. Now what you do is you find the most similar pair in the first step. Say blue and red. You line these 2. And the next one, you find the next most similar pair. Green and black, and align those 2. Now what you do is you align these pairs. So essentially that gives you an alignment of pairs, that gives you an alignment of pairs, and that gives you an alignment of pairs, which still is a pair alignment. Okay? And that essentially is the hack. There are different ways in which you can do it. You could now, in the second step, already put the next most similar into that one. So rather than doing black and green and then do a pair of pairs, you could immediately do the first pair and then see that this, in fact, the pair of pairs is most similar to green, so pull the green in then and build up like that. In principle, the idea is the same. It's just a different way of doing it. In the next lecture, which will be on Tuesday the 13th, I will continue on alignments, and on multiple alignments in particular, profile-based alignments. Thank you for today.